कोई चीज को तुम professor Nikita Dudla
should I ask directly पूजा क्या हो रहा है, क्या सोच रहे, ऐसा शादी के बारे में आप क्या सोच रहे हैं, ऐसे पूछना है और जनरल पूछना है?
थोड़ा सा घुमा करके
Good evening, one and all. My name is Dr. Bimal Chaudhary. I am working as a veterinary surgeon in the Department of Animal Husbandry, Government of Odisha, and also I am assigned as the National Youth President of Indian Veterinary Association. Being the organizing secretary of this workshop, this is our second day. Yesterday we had an off day because of this Diwali. So today we are going to start this lecture series. So today, may I invite today's moderator, Professor Dr. Santwana Pillai. Dr. Santwana Pillai is working as an assistant professor in the Department of Pharmacology and Toxicology in the College of Veterinary Science and Animal Husbandry, OUAT. She has been, instrumental in making this ethno-veterinary practice and implementation of it in the field level also. She has been guiding our students in the PG students and the PhD students also. I will request Professor Santwana Pillai to be the moderator of today's session. On behalf of Indian Veterinary Association, I invite all the participants and our two revered speakers, Professor Dr. P. Bambang, who is a professor of pathology in Indonesia, and also he is the General Secretary of our FAVA, Federation of Asian Veterinary Associations. I also invite Shruti Rastogi Madam. She has been working with the Indian Pharmacopoeia Commission since last 11 years. I request the moderator, Professor Dr. Santwana Pillai, to take this session forward.
Thank you, Dr. Bimal.
Thank you.
Am I audible?
Yeah, you are audible.
Good evening, one and all present all here in today's webinar. I am Dr. Santwana Pillai, working as Assistant Professor in Department of Veterinary Pharmacology and Toxicology, College of Veterinary Science and Animal Husbandry, OUAT, Odisha, India. Today, we all are assembled here online for the International Online Workshop of Veterinary Ayurveda. In the lecture series on globalization of veterinary Ayurveda, today we have two eminent speakers, James in this line, Dr. P. Bambang, Professor, Indonesia, and Shruti Rastogi ma'am, Scientist, Indian Pharmacopoeia Commission. I would like to start the program with a very brief introduction of Dr. P. Bambang. He is a professor of veterinary pathology. He is Secretary General of the Federation of Asian Veterinary Association, FAVA. He is Head of Veterinary Pathology Division, School of Veterinary Medicine and Biomedical Sciences, SBMBS, IPB, University. He is also the Chairman of Animal Biomedical Sciences, Master of Science Graduate Study Program, School of Veterinary Medicine and Biomedical Sciences, SBMBS, IPB University. Sir, I welcome you on the behalf of IVA and the audience present over here to deliver your speech. For the knowledge of the audience, I would like to say that Dr. P. Bambam will deliver his speech on efficacy assay of some plant extract for animal diseases. Sir, please.
Thank you, moderator, Dr. Santwana Pillai. Good evening, everyone. Hello from Indonesia. Now here is 8:40, about 8:40, but it's okay. It's okay for me. So today I'm very happy to be here to meet all of you, and we can discuss and also sharing information about the herbal medicine. So I would like to start my presentation, and hopefully what I want to present is very useful for all of us here. I want to share the screen. Okay. Can you see it?
Yes, sir, it's visible, sir.
Okay, thank you. So today my presentation topic is efficacy assay of some plants extract for animal diseases. In this opportunity, I would like to talk about 3 diseases that we are studied with herbal medicine. So I'm the professor in pathology, and also I'm the researcher in Tropical Biopharmaceutical Research Center at IPB University, and also the member of Board of Ethics, Indonesian Animal Drug Association. And I also a reviewer on the Indonesian Food and Drug Authority in the Indonesian government. Okay, so we start as an introduction. This is the classical animal disease divided into infectious, and we call non-infectious, like metabolic or degenerative diseases. For infectious disease, for example, like viral disease, FMD, AI, BRSV, and bacteria like colibacillosis, or fungi, candidiasis, and parasitic like coccidiosis in chicken. And non-infectious disease, for example, diabetes, cardiomyopathy, aging, and also tumor, for example. From all numbers of disease, we can say, for example, 4 of them. So medicinal plants, Indonesia is the world's 2nd largest of mega diversity in plant and animals. We have not less than 30,000 of plant species, where out of them 700 species are potential as herbal medicines. Medicinal herbs in Indonesia by 326 manufacturers. I believe it's more than, because this 2 or 3 years ago, I used not less than 180 medicinal and aromatic plants. So it's mean that we have very good potential for the medicinal plants. And the total raw materials consumption annually, not less than 10,223 tons annually consumption of these raw materials. And some of medicinal plants and all animal that we studied, for example, Curcuma santoriza, Curcuma aeruginosa, Phyllanthus nidori, Andrographis paniculata, Tiponium falciforme, Amomum compactum, Curcuma domestica, Momordica carantia, Singiber onypsiale, Lufas cindrica, and others. We have a lot of example for this, but this is just an example. And from the lower animal studies, we study also like earthworm, red mollusk, marine sponges, and others. This is what we studied, for example, concerning the herbal and also the lower animal. First, I will focus on 3 what we have studied. First is immunostimulant of antiviral effects of some medicinal plants prospecting for anti-avian influenza. We studied this due to the outbreak of avian influenza in our country, and then government are looking for the new drugs or potentially herb medicine of medicinal plants to cure this avian influenza. So in this case, we use Curcuma santoriza, Phyllanthus nidori, Andrographis paniculata, and Curcuma aeruginosa. We also working with the private company, Charon Popan from Thailand. So parameters we observe for these studies, immunostimulant activities in vivo, like antioxidant activity, toxicity, white blood cell count, antibody titers, phagocytic activities, histopathology, growth performance, challenge test to AI infection using a street virus, and also palatability when we exposure to the chicken, because we hope we can use this for the commercial feed. 4 group of chicken layers, 14 weeks old, and broiler from we start from the day old chick. We have 4 groups. unvaccinated group without plant extract, unvaccinated chicken plus plant extract, vaccinated plus without extract, and vaccinated chicken plus the extract plant. So we divided into 4 groups. And after treatment, we challenge all the chicken with the AI street virus, isolate from field in the BSL-3 facilities, because this is a zoonotic disease, so we have to be careful. And palatability tests, we use 300 heads of layer and 300 heads of broiler, and then we divided into 6 groups. Layer feed with commercial feed, layer feed with formula 4 and 7. We formalize the 4 herbs into several formulae. Actually, we start with 100 plants. We're looking for the potential plants. Then from 100 plants, we screening to 15, and then we deeply screening, and we found 4 plants. And for 4 plants, we formulate into several, and we found that formula 4 and 7 are the potential plant because of the antioxidant activity. Feed was given according to the standard of age, as is commercial farm. Drinking water ad libitum and quantity of feed consumption was measured weekly for 5 weeks. As the result, show that from 14 formula, as I said, from 4 plants, we have a 14 formula. We tested. We got 4 formula. Formula 4, 5, 7, and 9 show a higher antioxidant and toxicity activities. And then the formula 4 and 7 demonstrated a high potency for controlling avian flu based on the parameters responses. The challenge test of infection in the biocertificate level 3 for formula 4 and 7 show 40% as protected chicken from AI. It means then the chicken that not receive vaccinated, but we give the herb plants, and then we challenge with the AI virus treat, they're still alive until the challenge time finish. Toxipathological assays show that the herbs formula are relatively non-toxic to the chicken. So this is the data, what we assay: bioactivity of the antioxidant and toxicity of the plants, white blood cell counts, titer of antibody against AI, and phagocytosis activity and capacity. We also assay using the peritoneal macrophage, and we infected with the Staphylococcus non-A without A protein as the antiphagocytic. And this is the challenge test. You can see that unvaccinated without plants, 100% are died. Unvaccinated plus plants, about 40 to 39 still alive. Vaccinated without plants, almost 100%. This is one is traumatic, so something. Then almost 100% are still alive because they're vaccinated, and vaccinated with plants, 100% alive. Palatability test, most of the tested formula 4 and 7 are accepted by the chicken, because more than 95%, one is 94, the feed consumption. And we also assay the toxicopathology by histological examination, and we found that there is no histopathology, no specific histopathological lesions in all organs. As a conclusion for the first study, the formula 4 and 7 demonstrated a high immunostimulant effect and prospecting for the controlling as an anti-avian influenza in chicken. So we are collaborate with chicken feed company to use our formula in the feed for controlling the AI. The second study is about the herbs extract inhibit the bovine respiratory syncytial virus. We mimic the COVID-19 using the respiratory virus, but in bovine. We collaborate with the Japanese university, our collaborator, and also some of the from Japan and also from Thailand and from Indonesia. The material of the and methods we use, of course, the plants extract. We use 4 plants as the first for AI: Curcuma santorisa, Curcuma aerogenisa, Phyllanthus nirudi, and Andrographis paniculata. The cell and virus, we use A549 cell lines from adenocarcinoma of the alveolar basal epithelial cells, and also MDBK from the Martin-Darby bovine kidney. BRSV strain virus was isolated from Kyushu Island in Japan and propagated in the Faroe cell line, in the Faroe. Dilution of virus to achieve multiplicity of infection. And for virus yield reduction assay, we add the extract of the herbs using the DMSO, and we use DMSO as control. We treat it in 3 phases. First is pre-infection. The second is simultaneously infection, and the third is post-infection assay. So the cells, the cell lines were treated in these 3 ways. Viral RNA was then extracted from cell culture medium using a nucleocapsid nucleospin virus purification kit, or from cell lysate using a RNase mini kit. And then quantitative real PCR was performed. The data was then analyzed for all experiments using a triplicate of independent culture, and statistical of data was performed using one-way analysis of variance and also Turkey multiple comparison test. This is the cytotoxic assay. We give the cells for the with the extract, and we assay until day 6. Yeah, the cell seeding, several dose of extract, and then we assay the viability of the cells. And the antiviral assay, as I said, the herbs extracted were used at 2-fold serial dilution, beginning with the calculated safety concentration. This is the graph, the chart. This is the cell seeding, day -2, and then day -1. We for the pre-infection, we expose to the herb extract, and then for the D0, we infected the cell culture with the virus and let them to 6 days until we assay the antiviral. For the simultaneously at the day 0, we expose the cell culture together with the extract and the virus in the same day. And for the post, we add the extract after the infection until day 6, until day 5. And at day 6, we assay the antiviral activity. So this is the result, determination of cytotoxicity. So we found that, we have to abbreviate with P, Q, and R concentration here. We have 3 concentration at 2 hours, 44 hours, and 120 hours. And the interesting one, this is the culture of the cell infected with the virus, and this is the prey. So we infected the cell before we add the extract of the herbs, and this is on time together, and this is a post infection. We add the extract. Can you see that in herbs D, the pre-infection, the plant D is very effective in pre-infection states. But for the plant A, it is effective when we add in the one times together between the infection and the extract. And for the herbs B, this the plaque of the cell was still okay on the post-infection. So it means that the virus was inhibited after the infection of the cell by the virus. This is the same. So this is the selective index. We can see here herbs A, B, C, the CC50, IC, and selective index. This is the simultan, this is the pre-treatment, simultan, and post-treatment. And this is the plaque formation. You can see the from herbs, 4 herbs, and then we can see the supernatan and the cellulars, the inhibitory effect of herb extract to BSRV replication and plaque formation. So you can see there is a activity and also the number of cells and also the virus. As the conclusion, the 3 herbs of extract, even we use 4, 1 extract, it's look like not effective, have a strong antiviral activity against BRSV and low toxicity. The herb extract suppress BRSV activity by 3 patterns, which depending on the timing of the treatment. First is the inhibition of virus entry, the second of inhibition of virus propagation, and the third is inhibition of the virus release. The underlying antiviral mechanisms remains unclear because we need further studied about this. This herb extract could be safe and effective agents for preventing and treating bovine respiratory disease complex or BRDC. That's one is causing by the virus of BRSV. And of course, there is a correlation with the Pasteurella multocida infection. BRSV infection enhances bacterial adherence to the respiratory epithelial cells. This herb could help minimize the use of antibiotics in bovine respiratory disease complex, which will help prevent the global antimicrobial resistance. So this is the conclusion for the second study. And the third study I would like to share with you about the in vitro and in vivo anti-proliferation, anti-angiogenesis, anti-infection activities of Typhonium flagelliform extract on tumor cells. So this is on tumor cells. The method we use anti-proliferative assay, anti-angiogenesis in vitro and in ovo using a fertilized egg, in vivo anti-tumor activity in mice inducted by the DMBA as a carcinogenic agent, anti-invasion metastasis using the agar soft agar system, histopathology as the general examination, and we also use immunohistochemistry looking for what kind of pathways of this herb extract activity of this herb extract. This is the anti-proliferation assay. We can see we're using the MCAB1. This is the tumor cells from oral cavity of a dog. We developed to a cell lines. And the second is our cell lines also. We developed these cell lines origin from the mammary gland tumor of a dog. So we can see here the extract concentration. there is an activity of the extract, and this is the control. We use doxorubicin as the anti-cancer, and this is in MCMB2 cell. The extract was the activity was higher in MCAB1 compared to the MCMB2 cell lines, even they are both tumor cells. And for anti-angiogenesis, we use also in vitro assay using endothelial rat endothelial cell culture, and in ovo using the fertilized egg, we assay the development of blood vessel. This is the result. You can see also we adding the extract combined with the natural interferon for canine and also for feline. But this is, we can see here, the extract also give the anti-proliferation activity. And in ovo, this is for anti-angiogenesis. You can see this is without negative control. This negative control. This is using compared with interferon. And this is the extract of flagellum herb extract. The formation of the blood vessel are reduced. It means that they have anti-angiogenesis activity. And for the in vivo, we use a mouse. We induce with the DMDA, and this is the exposure time of the DMDA. And then we measure the tumor development in the back skin of the mice. And this is the histopathological. We identified the tumors. We have 3 tumors, anaplastic carcinoma, fibrosarcoma, and squamous cell carcinoma. So these 3 type cells are developed from the induction of the MBR. And we use these 2 antibody, the caspase-3 and PCNA, proliferating cell nuclear antigen. Caspase-3, we are looking for the apoptosis pathways, and proliferating cell nuclear antigen, we want to know about how the extract inhibit the proliferation of the cell. Caspase-3 and PCNA. And this is anti-invasion assay using agar soft gel invasion assay. And we will see the cell cannot invade to the inner layer of the soft agar gel. And we use 2 agar gel and collagen gel matrix for the invasion assay. And we found that the cell, they cannot invade more deeply when we add the agar gel with the extract of the medicine herbs. As the conclusion, the ethanol extract of Typhonium flagelliforme has an anti-proliferation of tumor cells in vitro, anti-angiogenesis, anti-invasion, and anti-tumor activities. They reduce the diameter of the tumor in induced tumor in mice. The mechanism of its anti-tumor is seen through apoptosis pathways, because we only use the apoptosis pathway. Of course, there is a lot of pathways on how the anti-tumor substances are preventing from inhibition from proliferation of tumor cells. So we just looking at the moment, we're looking from the apoptotic pathway. The ethanol extract of Typhonium flagelliforme can develop as a candidate of anti-tumor substances. Of course, we need further research should be performed to ensure and clarify of these mechanisms. So the take-home message, as you know, that Indonesia is a country with a mega diversity of flora and fauna and needs to be developed to maximize the utilization of the source, because we not yet maximize. So we have a chance to maximize this potentials of the medicinal plants. Medicinal plants or natural substances has a potency to be developed and used in the treatment of animal and human diseases. Research on medicinal plants or natural substances is still open for the purposes of developing new drugs for veterinary and human health. And synergy of many sectors is needed to develop, produce, and utilize the natural drugs in veterinary and human health. And so this is my presentation. I would like to thank to Director General of Higher Education for the grant to my team, and also to GSPS of Japan, to Center of Animal Disease Control, the University of Miyazaki, Japan, and the Tropical Biopharmaceutical Center, IPB University, and also the Research Center for Herbal Medicine, the Center of Agriculture Research and Development, the Ministry of Agriculture. And I very like Mahatma Gandhi, because his wise word, the greatness of a nation and its moral progress can be judged by the way its animals are treated. So this is very wise word, and I always keep in my mind. So thank you very much for the short presentation. Hopefully my presentation will give you a little bit more information about the medicinal plants. Thank you.
Thank you, sir. Thank you, sir, for your technical speech on the bioactivities of plant extracts. Your speech clarifies us on the immunostimulant and antiviral effects on bovine respiratory syncytial virus and avian influenza virus. Also, your antiproliferative and anti-angiogenic effect on tumor cells was very clear. The results of in vitro and in vivo studies truly gives a clear picture of the bioactivities of those medicinal herbs. It will encourage our audience in knowing the biomedical approaches for this. Truly, sir, it was a very nice presentation. Thank you, sir.
Thank you.
Thank you, sir. Next, I will, sir, should we take the questions now? I will request audience, if they have any questions, they can ask. They can put in the chat box or they can ask.
Good evening, sir. I am Dr. Surasmita. Am I audible?
Yes, you are audible, Surasmita.
Yeah. Sir, can you tell us about the herbal preparation that you used in avian influenza infection?
Sir, you are muted.
Sir, you are muted, sir.
Sorry, still muted. Okay. Yeah, as I explained to you, we start with 100 of plants. We try to find, because we have a lot of plants. Then we screening for 15 plants. And then we got 4 plants that are suitable for the research, for the study. Then we extracted using the ethanol extract as usual, as an extractant. Then we use the extract. Of course, this is still the whole extract, and some of them we already analyzed the chemical compound, but I'm not reported here, and what the active substances. Then we assay the activity of this plant extract. Of course, we formalize to 14 formulae for the extract. So we combine plant A, plant B, plant C, plant D with several portion, with different portion. And then we assay for the antioxidant activity. Then we got that 2 formulas are very promising. Then we use for the further examination using in vitro and also in ovo and in vivo directly to the chicken for example, anti-AI. We add, we giving the extract to the feed. We mix with the feed, and then we give the chicken for several days, and then after finish the experiment time, we assay all the parameters, and some of chicken we still remain, and we challenge with the street virus or AI street virus. We isolate it from the field that is very virulent in the BSLT facilities of the animal laboratory. And we received that 40% are still alive, even without vaccination of AI. We not give the vaccine to chicken, but we only give the extract. And when we challenge, 40% is still alive until the challenge test was finished. So this is my answer for the question of Dr. Surya Smita Raj. Is
Yeah,
this
thank
okay?
you, thank you, sir.
Okay,
Actually,
thank you.
I was wanted to know about that 4th and 7th preparation you used. No, you found
Yeah.
good result in that.
Yes,
I want to know.
only 4 and 7 for formulation. So I'm sorry, I cannot say clearly because still on the study. So hopefully after we receive the good finished, and then of course we are looking for the commercialization. Thank you.
Thank you, sir. Thank you, sir.
Sir, I think in Indonesia, vaccination against artificial influenza is legalized.
Yes, legalized.
In India, actually, we are following the protocol, and I think within 1 to 2 years, it will be legalized.
Yeah, this is our government policy to legalize, because it is spreading all the region of the country. So nothing else except we have to use the vaccine for prevention of the disease.
Thank you, sir.
Yeah, thank you.
Are there any more questions from the audience? Dr. Vimal, should we go to the next speaker, or we should?
Yes, we will go. We will not, because we have a little time with us.
Okay, thank you, sir. So now
Thank
we will.
you.
Thank you, sir. So now we will move to our next speaker, Shruti Rastogi, ma'am. She is going to deliver her speech on veterinary herbal medicine in Indian pharmacopoeia. Shruti Rastogi, ma'am, is an assistant scientist at the Indian Pharmacopoeia Commission. She received her master's in pharmacy from Amity University, Noida, India in 2011, and has more than 11 years of experience in the field of pharmacy. In 2011, Shruti ma'am began working in an analytical research and development at Panacea Biotech, where she was involved in the quality control of recombinant antibody-based biotherapeutics. She has many publications in highly indexed journals with good impact factors. Currently, she is working as a scientific assistant in Indian Pharmacopoeia Commission, Ministry of Health and Family Welfare, Government of India. Ma'am, I welcome you on the behalf of IVA and the audience present here to deliver your speech.
Thank you very much. Am I audible?
Yes, you are clearly audible.
Okay, and is my screen visible?
Yes, ma'am, it's visible.
Okay. Hello, good evening, all of you. Thank you very much, Professor Bambang. Actually, your presentation was very informative. Now, today, I would like to present something on veterinary herbals in Indian pharmacopoeia. So, this is a comprehensive presentation, and basically, it talks more of the veterinary products in Indian veterinary herbals in Indian pharmacopoeia. So, moving on to the slides. Giving a brief about IPC, that is Indian Pharmacopoeia Commission. Basically, this is an autonomous institute under Ministry of Health and Family Welfare, Government of India. It has basically a 3-tier structure. The structure you are, the image you are seeing in this picture, this is basically of the commission only, the building of the commission. And now IPC is having basically a 3-tier structure that is comprising of a journal body, governing body, and scientific body. And we also have many expert working groups, like we have approx 30 to 40 expert working groups with subject experts to guide on the standard setting. Moving on to the functions and mandate, like if someone asks what are the functions of IPC or Indian Pharmacopoeia Commission, the first and the primarily important function of IPC is to publish Indian Pharmacopoeia, the book of standards for drugs that are manufactured, marketed, and distributed in India. And unlike any other pharmacopoeia, means no other pharmacopoeia in India or maybe in other USP or BP, they're having a separate volume. So only Indian Pharmacopoeia is having a separate volume, volume 4 for veterinary drugs. It comprises of both chemical, then botanical, and vaccines. It's comprising of all the drugs in volume 4, separate volume for veterinary drugs. Then the second mandate is development of reference standards, that is IPRS you may call. Then the third mandate is publication of National Formulary of India. The latest edition was in 2021. Then the other function of IPC is to run the program called as Pharmacovigilance Program of India, that is PVPI. And basically, IPC is also a WHO collaborating center for pharmacovigilance and public health programs. Then the next function is skill development and trainings. We do continuous skill development programs and trainings for the academics, for the universities, for the students, and also for the industry persons. And the last function is national and international collaborations. Like recently, we have got a if any one of you are aware of Pharmacopoeia Discussion Group, that is PDG, that's an international group. And it comprises basically of 3 countries, the United States, Japan, and European Union. So now Indian Pharmacopoeia is also now being included in that group. And basically they are moving this as a pilot phase, and they examine our efforts. And if we complete their targets and all, will be included in the PDG group. Mainly good for all the pharma industry, as they can easily the monographs and the products can easily be harmonized. Moving on to the next slides. What's the monograph development process for IP? So the first of all, we select the monograph based on their inclusion criteria. Inclusion and exclusion criteria are there, on the basis of which the new monograph is selected. And then there is an acquisition of the specification of monographs, mainly maybe from any of the pharmacopoeia, or if any of the there are sponsors like manufacturers who give us the monograph. Then the IPC basically draft those monographs, and after public consultation, they publish it on the website, take consultation from the stakeholders, and then finalize it in the expert review committees. Further, if there is no revision required, then they are approved by the secretary cum scientific director, and they are published in IP. So basically, the inclusion and exclusion criteria for any of the drug for inclusion in IPI. Inclusion is like the drug should be in national health programs. It should be a part of national list of essential medicines. Then they should be approved by CDSU, and drugs should be considered appropriate by IPC. The exclusion criteria: drugs banned in India, they are excluded from IP. Obsolete drugs, and if any of the drug is considered inappropriate by IPC, IPC exclude those products from IP. Now, talking about the expert working groups of IP, we have various expert working groups. Like for basically for this audience, the expert working group of interest is herbal drug, a herbal expert working group, and veterinary products working group. Then what does IP standard or a IP comprises of it basically comprises of monographs from A to Z alphabets, and then we have monographs for herbals, we have monographs for biologicals, we have monographs for radiopharmaceuticals, we have monographs for veterinary products, and then there are also journal chapters. Now journal chapter basically contains the validated methods that can be used. Basically volume 1 is containing the journal chapters, and then we also have physical reference standards. There may be traceable patterns to demonstrate broad-based acceptability of procedures. Now talking on to the veterinary herbal medicines in IP, if we see the herbal medicines are being used by an increasing number of people. And why? Because they have no side effects or they have a minimum side effect. If we talk about Asian and African countries, we see that 80% of the people, like if you see the people in villages or people in remote areas, they today also prefer traditional medicines for primary healthcare needs. Then if we see herbal medicines are the most lucrative form of traditional medicines, as they generate billions of dollars in revenue, and India being a hub of these herbal medicines, basically, there are many of the herbal medicines that are being exported also from India. Now researchers look to traditional medicines as a guide to health, as 40% of the plant comprises key ingredients that can be used for prescription drugs. It is reported that herbal global market was worth rupees $151 billion in 2021, and Indian herbal industry is worth around 2.2 billion. So the global herbal medicine market is projected to grow from $165 billion in 2022 to $347 billion by 2029. So there will be a vast growth in the herbal medicines. Now, if you see how basically these herbal medicines can be used for veterinary purpose. Now, these herbal medicines can be used in the form of fresh herbs. These are basically chopped and mixed food. They are basically chopped and mixed with food. It is perhaps the ideal way to give herbs when they are available. Then they can be also used in the form of dried form. They can be administered directly to the food or making them infusions or decoctions. Then as alcoholic tinctures given directly or diluted in water and given orally carefully using syringe or dropper. Then as oil infusions or lotions, then also they can be used in the form of tablets and powders. If we see various herbal drugs that are present in IP and are being used for veterinary practice, I have consolidated a list of almost 45 of them. So basically, the first one comes here, Acacia nilotica, basically the Indian gum, and the part of plant used is seeds and bark, and basically it's used for acidity and disease. Then it's Athatotica vesica. Okay, now I just wish to tell, like, this is the list, and the indications are basically depending upon a review search. The review article is available for all of them, and I had just compiled all those indications into a single presentation. So these are various products that are in IP that can be easily quantified. Like what you can do is, if someone wants to test like any of these products, they can easily follow IP method. They are available in volume 3 of IP. So the test methodology, the macroscopic, microscopic evaluation, every test is present over there. You can easily perform the test from them and then use that for the specific indication. And then I talk about like recognition. Now, India has been recognized as a book of standards in mainly 4 countries, Ghana, Afghanistan, Nepal, and Mauritius. We are looking forward for more countries to accept our standards. And IPC is also having collaborations with many national and international organizations. Like we have a collaboration with EDQM, with the United States Pharmacopoeia, with British Pharmacopoeia, with development of herbal monographs, for impurity development, for DNA barcoding and reference standards, for academic trainings with WHO. Thank you.
Thank you, ma'am.
Any questions?
Okay, ma'am. Let me thank you for your speech. It was a very good generalization on the IPC functions and mandates, collaborations, the IP monograph development process, It really clarified on the veterinary herbal medicine uses and its roles and the administration one can do and the role of veterinary herbal medicine in global market. Really, veterinary herbal drugs has a future. Okay, ma'am. Now I will go to the audience. If they have any question, they can put in the chat box or they can say it directly also. Anyone from the audience, if they have any questions?
Yeah, good evening, ma'am. Can you tell us about that monograph?
Okay, ma'am. She wanted to know more about that monograph development process.
Okay. Basically, anyone, like anyone in India or anyone in globally can submit us the monograph. Now, what is the requirement for any of the monograph to be included in Indian Pharmacopoeia? Basically, that monograph should be approved by EDSU, the Central Drug Regulatory of India. And once that product is approved, then for a chemical product, our criteria is that it should have 3 to 4 manufacturers because there should be someone who can use those products. That's why there's a need to, we should develop a monograph. For veterinary, basically the herb or the veterinary veterinary herbal product, it should be of use. Like in if any of the product is currently widely be used, we suggest that you all should like submit us the monograph because veterinary I see like there is some gap in the veterinary industry and in the standards development. Maybe because the veterinary drugs that we are having in IP are there, they are very little as when we compare to chemical drugs. So we always suggest the stakeholders that you can submit us the monograph or you can send us the query. I just write the email ID of IPC on the chat box. You can please share your comments over the same. And I also mentioned the website over there so that you can easily mail us.
Okay, ma'am. Thank you. Anyone in the audience, if they have more questions, they can put. Or ma'am is putting her email. Okay, ma'am, there is a question. I think it's for you. Or is there any use of garlic for snake bite?
I'm sorry, I'm not aware of it. I'm so sorry. I'll just check and I can revert. No idea on that. If anyone from the audience can reply, then please.
Oh, I don't have experience with snake bite.
Sir, please. Sir, please, if you want to say something, you can.
But garlic usually we can use for skin parasite diseases in dog, like what you call the parasite.
Men's.
What do you mean?
Men's, you know, you called it a.
Yeah. Mine. Yeah. You're right. So just additional information. Thank you from me.
Dr. Bambang sir, another question is from the chat box. Is Cucurma longa having any broad spectral antiviral activity or it is specific?
Okay. Kurkumalong.
Yes, sir.
Sorry.
Yes, sir. Yes, sir.
Kurkumalonga, usually they have activity for hepatoprotector. Yeah, hepatoprotector, like Kurkuma santoriza. So this also have activity for this. But for virus, we not use this in our study. So this is my answer. Thank you.
Okay. Okay. Thank you, sir. Thank you, ma'am, for your nice speeches. Now, for the formal vote of thanks, I will request Dr. Bimal. Over to Dr. Bimal.
Yeah. Thank you all. Is Dr. Kumar present there?
Yeah, I'm here. Am I audible?
Dr. Kumar?
Yes.
Yeah. Yes.
Yeah, am I audible?
So let me invite. Yes, you are audible.
Yes. Okay.
Yeah.
Good evening, everyone.
Uh,
Myself, Dr. Kumar, here to give away the
So,
thanks for
Dr.
the two important speakers. Am I audible?
Yes, sir. Yes, sir. You are audible.
Yes, yes.
Yeah.
Great, great.
So Dr.
it's
Kumar
my
is a scientist at. Let me introduce first Dr. Kumar.
okay.
So Dr. Kumar is a scientist at Transdisciplinary University. He has been working since last 14 years on veterinary Ayurveda, what we call as in Sanskrit as Pasu Ayurveda. So he has been instrumental. He has developed a lot of formulas, and it is all available on their website of Transdisciplinary University and also the National Dairy Development Board. Not only the formulas, also the practical videos are also available. So you can go and see it also. So now I invite Professor Kumar to propose the vote of thanks and also speak something about the Pasu Ayurveda conclave that we are going to conduct in Goa. Welcome.
Okay, thank you, Dr. Bimalji. It's my pleasure and honor to be here to give away the thanks for the two speakers for the day. And good evening to everyone. First of all, I would like to thank Indian Veterinary Association to take this initiative to, start something on veterinary Ayurveda or Pashu Ayurveda on behalf of, veterinary fraternity to say, because it's been not addressed well so far in these years, where herbs have been major junk for drug development process through industry as well as through academics, but it is not been addressed. So with the help of Indian Veterinary Association, we could able to start this initiative as a webinar, series of webinar, as a part of our Lord Dhanvantari Jayanti. Lord Dhanvantari Jayanti was on 23rd October. So we celebrated as inaugural function on 23rd, and as a part of it, we are continuing this webinar series. I would like to thank both the speakers of the day. One is Professor Bombach ji, and given enough enlightenment on the process of research protocols carried at his university, and with the wonderful outcome of those results he has shared with us, and to say that how important to carry out the research for even for herbs, to say that how effective for its practical or clinical applications. Thank you so much for being with us, sir, and giving your insight on the protocol types and helping our participants to know more about herbs and its actions on birds. Thank you so much. And on behalf of IVA and on behalf of Ayurveda Foundation and TDU wholeheartedly, I thank you. We look forward to have more collaborations in future with you and your university, and we are happy to be a part of your work. We look forward, sir. Thank you so much.
Thank you.
Then the next speaker is our Dr. Shruti Rastogi madam on Indian pharmacopoeia. I'm very happy to know that in Indian pharmacopoeia, herbs have been entered and a lot of work has been addressed for the herbs for its action and its indication. I have more questions to ask, madam. So I thought personally I'll interact with the madam to know more about it. Thank you so much for sharing your insight and the way you are carrying out that Indian pharmacopoeia, not only for Indians and even for overseas, and you have a lot of collaborations to say. We are really enlightened to be for your lecture. Thank you so much for being with us and delivering an important lecture to understand more about herbs at Indian Pharmacopoeia. I thank all the participants who are on Zoom and who are on YouTube. So we look forward to have your more participation in the coming days with the webinar series. and you'll have different, stalwarts of different areas to be for whole week. And you'll get to know more about herbs and veterinary Ayurveda related work carried at different levels, to say from academics to industries, and from research orientation to field applications. This is what I can assure you all. You will have fruitful webinar series throughout the week, and I look forward for all your participation. And beyond that, I'll also would like to invite you all for important international conclave, which is going to happen in 10th December in Goa. Organized by Ministry of Ayush, Ayurveda Foundation, Transdisciplinary University, and Ministry of Animal Husbandry and Dairying Government of India. So we look forward all your participation in that event as well. So I think more will be shared through Indian Veterinary Association to you all through mails. I try to share with much of the for international and FAVA association members. And in the coming days, I'm going to share those details even in Indian Veterinary Association platforms. So thank you so much, and see you all again by tomorrow same time with 7 p.m. with the two important other stalwarts of Pashu Ayurveda and Veterinary Ayurveda. So I thank, and we look forward to have more such webinar series.
And it will be injustice if I will not, will give the vote of thanks to our moderator.
Oh, sorry, I forgot. Thank you, madam. Thank you, Santwana, madam. I'm very sorry. So thank you so much for moderating the whole event and keeping the time and everything. So we are again, a bit early rather than like other day, inaugural day, it was went 2 hours delayed by our event. So, but today it's not so like, you have finished in time. Thank you so much moderating and keeping, timely placed agendas for us. Thank you.
Thank you all. So with permission of our sir, I am just closing the meeting, sir. Thank you, sir.
Thank you.
Thank you so much. Yeah.
Thank you, madam.